This is the anti-aging guru Brian Johnson, who spends over $2 million yearly on the Blueprint Project with guidance from more than 30 doctors to conquer and reverse aging. When it comes to fighting aging, biological age is an important way to monitor progress. So, how does biological age reflect aging and how is it calculated? Hello everyone, welcome to WeGene. Today, let's talk about how to scientifically calculate aging. Aging is a complex biological process in which an organism's physiological functions gradually decline over time, its ability to adapt to the environment worsens, and its susceptibility to disease increases. And chronological age is the most straightforward way people measure aging. For example, my 78-year-old brother-in-law is obviously older than I was at 18. But in real life, among elderly people of the same age, some can spin like tops in the park, while others can't even take care of themselves. Relying solely on chronological age doesn't do a good job of explaining an individual's actual aging condition. To address this shortcoming, British scientist Comfort published a groundbreaking study in 1969, proposing a method to measure the rate of human aging through a combination of multiple tests. It's like trying to calculate how hard it is to get a train ticket during the Spring Festival rush by using the ticket purchase rate of passengers already on the train, while ignoring the fact that everyone on board has already managed to buy a ticket. Comfort's assessment method was similarly abstract. He used visual field and pupil diameter as variables to calculate age, but overlooked the fact that the visual field is inherently affected by pupil size. However, despite the limitations of his method, it opened up new avenues for later research on aging. People no longer measured aging solely by calendar age, but began to explore ways to calculate biological age. As science has advanced, our understanding of aging has also continued to deepen. These are the 14 characteristics of aging that scientists have identified so far, with the 4 colors representing 4 different categories. Take telomere attrition, which many people are familiar with, as an example. In 1990, scientists including Harley discovered a special DNA protective structure at the ends of chromosomes called telomeres. Telomeres gradually shorten as cells divide, and once they become too short, the cell stops dividing and enters a state of aging. Based on this, scientists have developed various methods to measure telomere length, which help assess the degree of cellular aging and, in turn, calculate biological age. Of course, this calculation method is not without its drawbacks. For example, some studies have found that athletes who participate in long-distance endurance trail running show a statistically significant shortening of their telomeres. In addition, factors such as chronic stress, smoking, and diseases can also affect telomeres, and even the rate of telomere shortening varies between different tissues. Therefore, telomere attrition is actually difficult to use as a universal standard for assessing aging. Besides measuring telomere length, there are actually many other methods for calculating biological age. Here is a table that I've put together summarizing the characteristics of several common biological age calculation methods. As you can see, each calculation method has its own advantages and disadvantages. In order to find a more reliable way to calculate biological age, a review published in a subjournal of The Lancet in 2017 systematically evaluated 6 biological age prediction methods. For example, in this evaluation chart, you can see that although telomere length has the most extensive foundation of validation studies, its ability to predict biological age is actually relatively weak. On the other hand, the method with relatively more thorough research validation and better predictive ability for biological age is actually the epigenetic clock, that is, DNA methylation. Moreover, cutting-edge preprint studies have shown that using DNA methylation testing, it is possible to build aging assessment models for various physiological systems, including the heart, liver, brain, lungs, and kidneys, and the predicted degree of aging in these systems is highly correlated with the risk of age-related diseases. Even the telomere length we mentioned earlier can be measured through DNA methylation testing. In fact, compared to traditional telomere measurement methods, telomere assessment using DNA methylation has smaller measurement errors, higher consistency, and greater accuracy in predicting cellular aging and health risks. Therefore, compared to aging assessment methods that rely solely on telomere length, which can only reflect a single aspect of aging, DNA methylation testing is clearly a more comprehensive and scientific way to measure aging. But after all this, what exactly is DNA methylation? Simply put, DNA methylation acts like a switch on DNA. When a methyl group is added to a cytosine C, in a DNA sequence where C is followed by a guanine G, it can turn a gene on or off without changing the actual DNA sequence. The methylation patterns on DNA change as we age. When the expression of related genes is altered, it can affect cell function and the aging process. For example, the anti-aging guru Brian, whom I mentioned at the beginning, has been continuously tracking his aging rate by testing his own DNA methylation. From 0.76 in 2022 to 0.64 in 2024. That's like aging only 7.6 months over the course of an entire year. You might be really curious, how exactly is such a precise aging rate measured through DNA methylation? This is a DNA methylation chip, and each square on it contains millions of microbads. Each microbead is coded with a large number of identical DNA probe sequences, and for each CPG site, there are multiple duplicate copies of the corresponding microbads on the chip. After the sample DNA is pretreated and amplified, it then hybridizes with the complementary probes on the microbads of the chip. The detection results at these sites are reported through fluorescent signals. With the help of a high-throughput chip scanner and dual-color fluorescent signals, we can distinguish between methylated and unmethylated states and quantitatively measure the methylation level at each CPG site. After obtaining the raw DNA methylation data, it still needs to be analyzed using various epigenetic clock models. By optimizing parameters through cross-validation, the testing company can finally produce an interpretive report like this, which includes the same type of aging acceleration factor as Brian's biological age and the aging status of various organs in the body. Of course, these tests and analyses are not services exclusively for the wealthy. In fact, thanks to advances in national technology, DNA methylation testing has already been successfully commercialized. Even the previously daunting invasive blood sampling has actually improved significantly. For example, the Yueling Bio Plus aging test, which has moved from the lab into thousands of households, uses the domestically produced BGI Bluomics bionic sampler. It can provide you with an almost painless sampling experience, and the process is so simple that ordinary people can easily complete it at home by following the instructions. Powered by a domestic data analysis system, it avoids costly overseas patent fees. Now, this comprehensive Yueling test, covering 270,000 methylation sites and 35 report items across 5 categories, is available for just over $270. And that's a wrap for this video. I'm Jeff, destined to either become a spinning grandpa in the park or a helpless old man in bed. Catch you next time.